I'm Jonah Comstock, Editor-in-Chief at pharmaphorum. I'm here at the AAPS National Biotech Conference in Boston, and I'm joined by Dr. Dhaval Shah, a professor of pharmaceutical sciences at the University of Buffalo. Thanks for joining me.
Thanks for having me.
So you are here, you're sort of an expert and an academic. You previously worked with Pfizer, and you're here to talk about simulation and modeling in biologics, right?
Yes.
So tell me a little bit about kind of your presentation today and what you're here to talk about.
So I'm here to talk about really the new kind of drug modalities, gene therapies, antibody-based therapies, which are really evolving very fast. And really how one can use the principles of pharmacokinetics and pharmacodynamics, which is basically how the body handles a drug and how drug reacts within the body, and also mathematical modeling to actually make informed decision regarding optimization of the drug molecule, their preclinical to clinical translation, and then optimization of the dosing regimen within the clinic.
And what's the current state of the art? I mean, are people doing this now to some extent, and they could be doing more of it, or is this really something that needs to kind of have more uptake?
So I think this field has evolved very fast as well. Of course, we learned a lot of the principles from small molecule drugs, which are, of course, very important as well. But over the time, we have evolved these models to account for biological behavior of biologics. Now, state of the art right now still doesn't remain as good as one would like because of the lack of data. In this day and age where everybody wants to make an AI model of everything, what we're missing in our field is data that you can train these models on. And we use some of AI model, but most of them are deterministic models using a differential equation. But even with the system small, the problem has been is that we are not having enough data. So I think we are limited by the data of the novel drugs within the cell-based system, within the animals, and even the clinical situation of different scenario to help us make the best of the mathematical models. So there's still a lot of room for us to grow.
In a perfect world, when would you imagine the models would come into play in terms of where along the drug development, drug discovery timeline?
Ideally, you should have these models before even you make a drug molecule for a disease. We typically like to know that for many diseases because we make systems-based model. For example, let's talk about oncology and a cancer, right? Where basically you may know that how cancer cells are different than normal cells. You can make a mathematical model of the system or the molecular pathway that the cancer has. And then you can use model to perturb the system and say, this is the best targets. And then you can make a drug molecule which binds to the target. But then which drug to make? Are you going to make antibody drug conjugate, gene therapies? Well, there you can utilize different kind of models for the disposition of the drug and say, well, with this dose, you can get to this target at good enough concentration without toxicity, and you should pursue this. So ideally, we are hoping that we'll have a framework that one can use before even start making the drug. Because when you start making a molecule, you're already into $1 million into the hole because you have to make the molecule, do the studies. And ideally, we would like to have this in silico model applied much more prior to starting the wet lab process.
And of course, you have to be able to really trust those models, and so that you have to verify them over and over again and see that the actual molecules behave the way the models did, right?
The proof is in the pudding, and that's exactly is the same. So one of the things that distinguishes our lab from others is that we have wet lab and in silico component. People in my lab work excruciatingly hard to really generate novel data that nobody's ever generated, and then use that to make the model. Precisely because, as you mentioned, we only like to make the model that we can basically trust does a priori. And that's where my experience with Pfizer comes into picture, where I realized making the model and publishing the paper is the beginning, not the end goal of an effort. Once you trust these models to make decisions, that's where the models are useful. And you are right, absolutely right. You should be able to trust them, and data is needed, and you need to have both wet lab and in silico people talk to each other for that.
There was a lot of discussion yesterday about regulators and regulators not really trusting or respecting the in silico models yet. And so you have to reproduce it in vitro if you want to actually get something approved, even with a breakthrough designation. I mean, how do you think that is evolving, and how do you think it ought to?
So I think I would like to say, who are regulators, right? They are actually ex-students and ex-colleagues of mine and current friends of mine as well, right? They're scientists just like you. One would not really like to consider regulators are somebody sitting in a basement making decisions for you, right? If you show that the model is made using robust data, if you can actually perturb the system and show in various situations model simulations matches the data or prior prediction, they would trust you. only when you basically say, I have a model and now all the simulations I can do, you should trust without having any validation, even I wouldn't trust that. So I would say, I'll take that as a very case-by-case basis, but at the same time, give them enough credit because they want to make sure that these are reliable models before you can just do human studies, which nobody would like to do on.
Yeah. Yeah, I mean, it's always a balance, right, between speed of innovation and that safety and caution.
Yeah, being in this field and really leading several efforts, I'm really cognizant and really concerned about overselling of the model as well. Even the reports from FDA last month saying, we will get away without animal models and stuff like that. I think it's a bit hyperbole. There's not much evidence regarding that. So yeah, I think as you know, right now regulatory is also very much in flux. We're going to have new partners and new leadership over there, and we'll have to convince them basically as to the benefit of the model. At the same time, caution them towards just expecting AI models of everything, which is not going to happen, in my opinion, 5-10 years until now.
When we talk about simulation and modeling of a drug, what are we really saying? Is it just sort of like feeding in a lot of data and predicting what's going to happen, or are you actually creating a model of like the inside of the body of the relevant cells and sort of running a bunch of simulations and watching it happen?
Exactly like that. There are ways to visualize, but we don't visualize, we just look into outcome. So for example, monoclonal antibody-based models, right? At this point of time, we are able to simulate giving certain dose into the individual, predicting mathematically over the time how much drug will remain in the blood and different tissues. And then based on known tissue concentration, you are able to understand how much of the drug target will be engaged, and would that really lead to the efficacy or is suboptimal dose. At the same time, let's say your toxicity-based issue, how much a target engagement will happen there as well, and would you go over the threshold or not. So literally, as you said, we are able to simulate drug concentration all over the body and predict the target engagement. And then based on our understanding of how much target engagement is beneficial, how much is toxic, we can simulate those effects.
That's so interesting. I mean, in the real world, there are so many variables that affect how a drug impacts a person, and you're down to the kind of individual factors of who that patient is, what other drugs they might be on, what else is going on in their life. I mean, how can a model account for all that, or does it all sort of come out in the wash?
No, they do account for that, and we have terms for all of them. For example, different individuals have different properties, and we call them covariates. For example, it's known that body weight is a significant covariate. If you give the same flat dose, let's say 100 milligram, into 50 kilogram individual versus 100 kilogram, their concentration will be different. And we can actually simulate that as well. If we are taking another drug with that, we call them drug-drug interaction. You can also simulate that. So all the things you're envisioning are realistic, and we can actually make virtual patients and do clinical trial simulations to account for each of the scenario that you're interested in.
Wow. So yeah, so the modeling is very complex.
It is very complex, and it's becoming very detailed and system-oriented. And that's why, again, there is some truth behind trusting this in silico model for antibody-based drugs at least. And again, we have played a significant part into that as well. But hope is now gene therapies, antibody-drug conjugates, these novel drugs. Hopefully, we can do the same thing with the model that I'm going to present today.
That's awesome. And how much computational power does it require? I mean, is this something that's only become possible recently with the AI and computers where they are, as we've been able to do for a while?
It's a very good question. So I think systems-based models are becoming more and more complex. And in my lab, we have run the model that takes about 24 hours to simulate on a PC, and which is unattainable. So it depends on whether you have a software that can be parallelized. So in University at Buffalo, we have cluster computing and computer systems and supercomputers. So we're able to avail them. So, and again, AI models, we make certain AI models. They actually are even much more power hungry, and we need GPUs, which are expensive to rent as well, basically. But I would say computational powers is one thing, and then understanding the system is the second thing. An ability to understand the system and write them a differential equation and simulate, it's not as onerous on computational aspects. But when you want to, let's say, estimate a parameter that you are not aware of based on the data, or you want to simulate thousands of individuals based on different situations, that's where it becomes computationally really demanding. And when you put AI onto that, for example, we use a lot of neural network models, and we combine them with differential equation models, and estimating the weights of each of the nodes, and those takes time, and number of epoch that you want to run and things like that matters as well. So yeah, I would say again, of course, higher computational power is helpful, but that's not the only detriment to the field or limitation to the field so far. It's been just availability of the data and understanding the system as well.
I mean, you mentioned gene therapy in that. It sounds like that's an area where maybe the modeling is less sophisticated, but that's what you're working on.
Absolutely.
What makes that more challenging? Are you having to simulate almost like another level down at the genetic level instead of just the molecular level?
Yes and no. Yes, we'll have to, but I don't think that's the problem. The problem is the data. For example, CRISPR-Cas9, when Nobel Prize was awarded a few years ago, people said this is going to change the world, right? Basically, but we still don't know once you cut a gene, are you going to really be able to insert the gene of interest, or are there going to be random genes injected within them? So again, just lack of understanding of the system, not enough data at the system level is a limiting factor in my opinion. Once you do that, then once you know all the players involved, you can model them. So I think just knowing all the players involved in the pharmacology, and again, really there are, for example, adeno-associated virus-based gene therapies, they actually have certain issues with toxicity, immunogenicity, and able to understand the adverse effect as well, so that we can find balance the dose has also been a problem with gene therapy.
So let's talk about data a little bit before we wrap up. I mean, so much of it is siloed. So, it's proprietary, or it's not published because the result wasn't favorable. I mean, how do you source the data to build your models, and what do you think the industry could be doing better to make more data available to more people?
Yeah, I have a ideal perspective and I have a practical perspective because I have been to industry before. In this day and age where pharma is really going through tough time economically as well, I wouldn't expect everybody to spend millions of dollars generating the data and just really share. That's your proprietariness, right? So I understand why they are not shared widely. But ideally, I would love for each of them to make a repository, generate all the data. And I advocated in certain cases. We are actually a consortium known as Center for Protein Therapeutics at University of Buffalo. It's funded by top 10 pharmaceutical companies of the world. And they do come in and sometimes share the data non-competitive and actually help us to build the model. So I would say ideally, I would like for them to share non-proprietary data as the project gets terminated and they don't have interest in the pathway. But I understand why they wouldn't do it, because it costs, I run a wet lab, as I mentioned to you, and it's tremendously expensive to generate the data. Anybody can come in and say, hey, just share the data with me, and then we'll just use it. But the time and effort it took to generate the data, you should be given some of the, I would say, acknowledgement for that as well.
Yeah, so we need kind of systems and structures.
Incentives for just sharing the data, and basically a way to make sure that you're acknowledged and you're always connected with the data when it is used and rewarded for the generation of the data.
Anything we haven't talked about yet that you think is an important piece of the puzzle?
No, I would just say, again, understanding how these molecules work, novel therapy especially, understanding preclinically and within the ex vivo models, how they behave is very important. Most of the companies in a race to go to the clinic don't generate the real mechanistic data that is needed to understand the system, and then they're surprised in the clinic. And I would say, I understand also because it's cost money and time, but if you're committed to a pathway, they should take more effort to understand the pathways, and really maybe they can come up with backup molecule that will work if the first one doesn't, rather than just this one shot wonders and see what happens with that, and the company falls because one molecule doesn't work out.
Yeah, so you save money in the long run, even if
yes, absolutely. And I understand some of the business models are asset based, but if you really have a vision to have let's say a therapy for rare disease, or a modality that is really novel and you have IP on, I think it's worth generating data, use mathematical model to help you translate, rather than just do some experiment, go to the monkey and just cross your finger and say it's going to work out.
Awesome. Well, thank you so much for chatting with me. Learned a lot, and look forward to your talk.
Okay, thanks. Appreciate that.